Good morning, everyone. My name is Kodri Adewale. I'm a third-year PhD student in neuroscience at the Montreal Neurological Institute, and I'll be presenting our work on how we use the mathematical model to integrate gene expression and neuroimaging data to advance our understanding of healthy aging and Alzheimer's disease. So there are many, as we know, there are a couple of hypotheses about the underlying cause of Alzheimer's disease, and that also extends to aging. And in looking at this hypothesis, both okay, so both observational models, like the one using Jack et al., and data-driven models have been used. However, if we look at these models, they didn't really look at the interactions between these biological factors or processes. So, for example, abnormal beta amyloid deposition, tau deposition, or glucose hypometabolism. So, like, how does tau influence amyloid, or how does glucose metabolism influence cerebral blood flow? So, because of this, what we did in our model was to look at gene expression data and also a couple of neuroimaging modalities, and see how these factors or processes interact in driving aging or disease progression. So if we look at a macroscopic factor, so here now in when I say macroscopic factor, I'm looking at the neuroimaging modalities separate from the gene expression. So if you look at the macroscopic factors, if you look at the changes over time, either due to healthy aging or due to Alzheimer's disease progression, right? We say that these changes are caused by the interactions between different macroscopic factors as defined by the imaging modalities we can see here. And we believe that these interactions are influenced or modulated by gene expression. Let me hide this. Okay. That these interactions are modulated by gene expression in a particular brain region. And of course, also the alterations in these macroscopic factors that are coming from other brain regions. So if we want to explain this mathematically, what we are trying to say is that if we look at a particular brain region I, and we are looking at the changes in a particular macroscopic factor defined by imaging modality, any of these 6, over time, we are seeing that this is due to the interactions between all the 6 macroscopic factors in that particular brain region, defined as S here. And we believe that those interactions are influenced or modulated by the gene expression of all the genes in that particular brain region given by GIK. And this alpha is the strength of the modulation, or the strength of the modulating effect of the gene expression on the interactions between the macroscopic factors. And of course, the alteration in the particular macroscopic factor under consideration here that's happening in other brain regions, that alteration also flows into the particular brain region we are considering. And of course, that flow can be modeled by the connectivity matrix we see here. So in modeling the data, what we did was to use gene expression from Allen Human Brain Atlas across 138 brain regions. We also looked at 6 different imaging modalities, which define cerebral blood flow, beta amyloid deposition, resting for neuronal activity, glucose metabolism, atrophy, and tau deposition. And what we used the diffusion MRI for was to get the connectivity matrix. And we also looked at 5 different cognitive evaluations. So for the modeling proper, this represents the left side of the equation where we had ds/dt, just to give us an idea of how the modeling was done. So if we take a particular subject and look at a particular macroscopic factor, let's say beta amyloid now, and we say that's 1. Then we looked at the changes in that particular macroscopic factor over time in a particular brain region. And if we do that for the whole brain region under consideration, where we're having 138. So if we concatenate that with the 5 other microscopic factors, so we could actually build a matrix for a particular subject. And if we solve that, if we solve that matrix using any sparse regression model, but here what we did was to use Bayesian regression, we could obtain the alpha parameters that we saw in the equation that I defined earlier. And the alpha parameters will give us the idea of how much a particular gene influences the interaction of a particular macroscopic factor with other macroscopic factors in a particular brain region, right? And after doing that, we looked at the ability of the model to reconstruct the what's called the data that we fed into it. And as we can see, with disease progression, the model performed better. And we attributed this to the fact that as you go into the later stages of the AD spectrum, the alterations or the changes in those imaging modalities, so the macroscopic factors we are talking about, seems to be larger, and that gives the model more information to optimize. So the next thing we did was to divide the subjects into two. So we look at subjects that were diagnosed as healthy control at baseline, and these subjects did not convert to either MCI or Alzheimer's disease over a period of 8 years on average. So we use those subjects to model LV aging. We also looked at subjects that were diagnosed with probable AD, Alzheimer's disease at baseline, and those that were diagnosed with those are considered as LV controls or cognitively impaired subjects, but these subjects later converted into Alzheimer's disease. So we use those subjects to model Alzheimer's disease as so the distribution of the subjects can be found here. So what we did was we actually did this for both LD aging and Alzheimer's disease, but I will use LD aging as a case study. So if we combine the alpha parameters that we obtained after applying regression on the data from each subject, so if you combine those alpha parameters on the left side here, and we also calculated the slopes of the 5 cognitive evaluations that we considered, then we applied singular value decomposition to look at the cause correlation between the parameters defining the interactions between the genes and the imaging modalities that we considered with the cognitive scores. And after doing that, just to evaluate how stable this analysis is, we did 10,000 bootstraps. And after that, we found out that the first principal component was the only stable one, and that accounted for about 50% of the covariance or the cause correlation between the left side and the right side there. Then for Alzheimer's disease, the variance explained was about 68%. So the next thing we did was to then calculate the bootstrap ratio that we obtained from the bootstrapping, and this enabled us to identify those genes that were highly contributing to the variance explained by the first principal component. And the bootstrap ratio was obtained by dividing the saliencies, which is the actual contribution before shuffling, and dividing that by the standard error obtained from the bootstrapping. And as we can see, we identified 8 genes driving LV aging and about 211 genes underlying Alzheimer's disease. So the novelty of this model is that it doesn't just tell us which genes are driving LV aging or Alzheimer's disease, but it also gives us an insight into the particular biological process or macroscopic imaging modality that particular gene is interacting with or modulating. So for example, if you look at SESN1 gene, this particular gene has been reported to inhibit aging, to modulate exercise, and also to be involved in oxidative stress. So what we found out here was that it influences the interaction of cerebral blood flow with other 5 macroscopic factors that we considered in driving longitudinal change in neuronal activity. So we also obtained the same thing for Alzheimer's disease, and most of the genes that you can that you find there, most of them have been reported to be associated with Alzheimer's disease before. Another thing that we did was to compare. So we carried out a functional analysis where we looked at the pathways in which those genes that we identified for LDA gene and Alzheimer's disease are involved, and we found 4 pathways to have overlapped between LDA gene and Alzheimer's disease, and those were the pathway for apoptosis, which is programmed cell death, oxidative stress, G protein coupled receptor signaling, and immune or inflammatory response. We didn't find any overlapping gene between the two, except this LSM6, which is a gene that is involved in regulating gene expression and mRNA splicing. And a previous proteomic study has linked this particular gene to LDA gene in muscle cells, and also there are studies that associated dysregulation in mRNA splicing to Alzheimer's disease, but we haven't seen a study specifically identifying these genes and this gene and linking it to Alzheimer's. So in summary, we developed a mathematical model which allowed us to directly look at how gene expression modulates the spatial and temporal interactions of different imaging modalities in LDA gene and Alzheimer's disease. And most of the interactions that we found have been reported by previous animal studies. And the novel ones that we also discovered can be validated through experimental models. And looking at therapeutics, we've first, the optimization that we did allowed us to look at the iterations or the alterations in gene expression at individual level, and we believe that this could be helpful for personalized treatment. And of course, there are some theories that believe that the cure to Alzheimer's disease might lie in combining different therapies. So, for example, targeting tau and amyloid at the same time. So, if we know some genes that are highly influential, and we know the factors that these genes are modulating, right? So, if we have a particular gene that affects, let's say, glucose metabolism and amyloid, then instead of targeting amyloid and glucose metabolism, we could just target that gene, and that could serve as a replacement for combination therapy. Thank you for listening.
Okay, my name is Mustafa, and I'm a PhD student in the Animated Lab. First, I would like to thank NeuroSymposium committee to give me a talk. My talk today will focus about part of my PhD work, which concern about the role of mitochondrial antigen presentation in the establishment of Parkinsonism. Okay. What have been proven before is that BINC and Parkin, which are mitochondrial protein, two mitochondrial protein, play a crucial role in immune system. And upon mitochondrial stress, thanks to this two mitochondrial protein, damaged mitochondria can be cleared in a process named mitophagy. While in their dysfunction or absence, and upon mitochondrial stress, mitochondrial-derived vesicle can be formed that allow degradation of Ag compound of mitochondria. What Diana Matthews, my mentor, had been proven before is that there is a specific mitochondrial-derived vesicle that is formed upon recruitment of certain X9 and RAP proteins in absence of BINC or Parkin gene or protein. Sorry. The issue is that this mitochondrial-derived vesicle might later fuse with endosomal compartment, the issue that allow mitochondrial peptide to be presented on major histocompatibility class molecule at antigen presentation in a process named mitochondrial antigen presentation. That can lead to activation of T cell. This specific T cell, that is mitochondrial specific T cell, can later attack neurons that present mitochondrial peptide on its surface, leading to its degeneration. In case of targeting dopaminergic neuron, it might lead to progression of Parkinsonism. The issue is that, or one important experiment that needs to be done is to assess the role of mitochondrial antigen presentation and the role of this mitochondrial specific T cell in the establishment of BD-like symptoms. Okay, the story started when our team noticed that upon infection of Bink or Barkin deficient mice with bacteria, a specific mitochondrial-derived vesicles that contain OGDH. OGDH is a matrix mitochondrial enzyme, increase significantly in antigen-presenting cell. That has been shown to increase the mitochondrial antigen presentation fold change in vitro that can be detected by OGDH-specific hybridoma 2CZ in both in vitro and in vivo level in BINK-KO rather than wild type. By looking at CD8, they also noted that there is a significant increase in CD8 OGDH-specific CD8 T cell. This means that mitochondrial antigen presentation can lead to autoimmunity. But what needs to be explored is that mitochondrial-specific T cell were not directly assessed for their ability to target specific dopaminergic neuron in vivo. And we don't know what is the MITAB or mitochondrial antigen presentation in Parkinson's disease and what exactly is the phenotype of this cell. So in order to find that, we decided to study the role of mitochondrial specific T cell in vivo, and also to identify and characterize mitochondrial T cell to have visualization of this mitochondrial specific T cell phenotype. First aim can be achieved by answering question, are mitochondrial specific T cell able to induce BD-like symptom? And in order to answer this particular question and validate the hypothesis, we build a new mirror model in which we can recapitulate activation of mitochondrial antigen presentation and presence of mitochondrial specific T cell and their infiltration into the brain. Simply by injecting TCR transgenic interferon gamma positive CD8 T cell, which is OGDH specific CD8 T cell, and by injecting Brittus toxin, which break or make blood-brain barrier leaky, so this OGDH specific T cell can access the brain. So by this model, we enable directly assess the contribution of mitochondrial specific T cell that is generated by activation of mitochondrial antigen presentation in the generation of BD-like symptom. And after that, we assess or we validate the movement disorder via different assay, bull test, which is a reliable test to validate movement disorder by putting the mice upside down and count the time that the mice consume to descend the bull. Also, we do actimetry that tests both movement and behavioral disorder or movement behavioral activity of the mice. And finally, to check neurodegeneration via immunohisto or immunocytochemistry. What the bull test tells us is that after interferon gamma positive mitochondrial specific CD8 T cell injection that is obtained from 2C mice, when we inject in BINK-KO mice, we see a significant retardation after day 25 in the mice injected by this mitochondrial specific T cell. That can be reverted when we administrate L-DOPA treatment. But this can be seen in both BINK-KO and wild-type mice, which indicates that BINK1 deficiency is not involved in the killing of dopaminergic neuron. Moreover, to validate that the effect is specific to T cell injection and not from Brittus toxin, we compare Brittus toxin injected alone against Brittus toxin together with 2C, and we can find a significant difference between two injections starting from day 22. Moreover, The actimetry assay approved or validate the Bull test result, where we can see there is a significant difference in the total distance traveled by mice that is injected by this CD8 positive OGDH specific or mitochondrial specific T cell rather than saline injection, that increase in significant level when we add ambulatory distance together with a significant distance travel, sorry. Moreover, the movement time tend to be increased in this mitochondrial specific T cell injected mice, despite it didn't reach a significant level. And when we check the behavioral issue, we can see that stigmatotaxis, which is a behavioral index factor, is non-significant between easier OGDH specific or mitochondrial specific T cell and saline injected mice. So we went to validate dopaminergic neuron degeneration in brain section, and we can find that this mitochondrial specific T cell injected mice has a significantly low number of Th positive cell and density of this staining as well, which indicates that mitochondrial specific T cell can establish Parkinsonism. But despite that, what Diana Matthew saw before is that upon infection, bacterial infection of BINC1 KO, there is a significant increase in both movement time or time for descending the Bull test, actimetry and everything, and even for the degeneration of the dopaminergic neuron rather than this model. And that might be because we only inject interferon gamma positive CD8 T cell, which is not represent the same T cell phenotype that is present in BD patient. So we decide to check BD phenotype in BD patient, and this is done by my colleague Camille Michaud, and she finds that upon screening for mitochondrial specific T cell in peripheral blood mononuclear cell, she can validate that this mitochondrial specific T cell has a phenotype of Th1 and Th17 in the term of CD4 T cell and TC17 in the term of CD8 T cell. So in order to mimic what really happened in BD patient, and actually because we don't know yet whether mitochondrial antigen presentation is over the phenotype that are induced or not, so we tested what if the T cell is induced by antigen presenting cell. This antigen presenting cells that able to perform MITAB. So we decide to inject a stimulated dendritic cell derived from BINC-KO mice that had been proven before that it can perform significantly MITAB upon LBS induction. And then we check the phenotype of T cell after this injection of antigen presenting cell specifically perform MITAB by flow cytometry. And what we find is that the mitochondrial specific T cell has a form of Th1, Th17, and TC17. And this is the same phenotype of T cell we find in human. So in the future, we will use this model to study the induction of BD-like symptom in mice and neurodegeneration. And finally, we can conclude that BINC1 deficiency is not involved in the killing of dopaminergic neuron. In other terms, whenever the mitochondrial specific T cell is established, the genotype has a less significance in this story. And mitochondrial specific T cell present in Th1, Th17, and TC17 phenotype. And mitochondrial specific T cell induced BD-like symptom and dopaminergic neuron degeneration. And immunization with LBS treated BINC-KO DC induced mitochondrial specific T cell with the same phenotype that had been seen in BD patient. Finally, it will be also very interesting to study the viral model. It is a case for PB1F2 proteins that from influenza virus. That is a minor epitope that is only visible when the immunodominant epitope are deleted. It would be then interested to see if the protein is presented through the formation of MDV and what kind of signal induce the presentation and whether mitophagy inhibit its presentation or not. And I would like to thank my mentor, Dana Matthews and Martin Titro, my colleague, Camille, Rino, and Annie, and for sure our sponsor as well. Thank you.
Hi everyone, I'm Maxime Kuzik, and today we will discuss together how the activation of interleukin 1 beta is a crucial step in multiple sclerosis. So interleukin 1 beta is an autoimmune disease where the immune cell turn against the central nervous system of patients, causing various clinical symptoms depending on where the neurons are attacked, such as visual impairments, cognitive deficits, or even paralysis and pain. And also, it is a huge social and financial burden because around 80,000 people are affected with MS in Canada, and it is a disease diagnosed around 20 to 40 years of age. And most unfortunately, no cures are available against multiple sclerosis. So although it can be slowed down, the neurodegenerative symptoms can never be reversed. So what happens in the central nervous system during MS? So we have here immune cell that will cross the blood-brain barrier and attack the myelin sheath of neurons, causing axonal degradation and loss of symptoms that are translated with previously mentioned symptoms. And also we see here that interleukin 1 beta is involved in multiple steps of this process. And moreover, our laboratory has shown that without IL-1 beta, mice cannot develop the disease. Thus, it is very interesting to understand how this IL-1 beta is produced during MS in order to block its production. So IL-1 beta is produced through two main mechanisms. The first one that will lead to the transcription of the immature form of IL-1 beta, whereas the other signal will allow the formation of the inflammasome, a multi-complex, a multi-protein complex, composed of a receptor of inflammatory molecule, the immature form of caspase-1, and those are bound together by the adaptor protein ASC. And once the inflammasome is formed, it will activate caspase-1 that will in turn cleave the immature form of IL-1 beta into its mature and active form. Thus, we can see that ASC is a central protein which allows the formation of the inflammasome and production of active IL-1 beta. Thus, we hypothesize that if mice do not have this adaptor protein ASC, well, the inflammasome will not be able to be formed, so IL-1 beta will not be activated and mice would be protected against the disease. And to verify that, we will induce a disease in mice genetically engineered to be lacking the ASC adapter protein. And then we will analyze how the disease progresses in these mice. And also we will check their immune cell using proteomic and bioinformatic analysis to understand how IL-1 beta is produced. So the mice have a disease called experimental autoimmune encephalomyelitis, or EAE, which mimics multiple sclerosis, and it is induced by two injections. The first one that will drive an immune response towards the central nervous system of the mice, and another injection that will promote this immune response. And the mice will develop a paralysis that begins at its tail and will progress to its head, where a clinical score, is addressed at each stage of the paralysis. So the clinical scoring indicates us the severity of the disease. So here we induce EE disease in mice lacking the adaptor protein ASC. So here we have the clinical scoring of EAE at different times following the induction of the disease, and we compared control mice with mice lacking the ASC protein. And we can see here that they developed the disease in a similar fashion than control mice. Thus, this indicates us that maybe cell types are involved and can produce IL-1 beta even if the ASC adapter protein is not there. Thus, we induced the disease once again in mice lacking ASC adapter protein, but that were also depleted in monocytes. And we see that these mice are completely protected against EAE. They do not develop any symptoms. Thus, this shows that monocytes can produce IL-1 beta even if ASC is not there. So we studied these cells more deeply in vitro, where here we see monocytes that were allowed to transmigrate across a membrane seeded with endothelial cell, which represents the transmigration of monocytes across the blood-brain barrier. And we analyzed the production of active IL-1 beta using Western blots. So here we have our monocytes lacking the ESC adapter protein. And first, we showed that when these monocytes are alone, they do not produce active IL-1 beta. However, when they are in presence of endothelial cells, they are able to produce active IL-1 beta, which means that this new interaction between monocytes lacking ESC adapter protein and endothelial cells brings into play new proteomic interactors that needs to be taken into account if you want to fully understand how IL-1 beta is activated. Thus, we try to identify these protein interactors using proteomic and bioinformatics. So here are the proteins that we found, and I know it is a very complicated figure, but I will walk you through this. So we analyze the proteins in the cytosol and in the supernatant of monocytes lacking the ESC adapter protein and we identify them using a mass spectrometer. And then we look at the literature to find proteins that are involved in the IL-1 beta activation pathway. And we can see all of these proteins here. So we analyze them using a string analysis, which shows which are the interaction between these proteins that are already known in the literature. So for example, we have here our caspase-1, and we have here IL-1 beta, and we know that that caspase-1 can activate IL-1 beta, so these two are linked here. However, we can see other interesting links. For example, caspase-3 here can also activate IL-1 beta, and it is not in the main inflammasome pathway. Also, we have other interesting interactions, such as cathepsin G, metalloproteinase 9, and cathepsin B that are linked to IL-1 beta and caspase-1. Thus, maybe these proteases can activate caspase-1 or IL-1 beta, and then lead to the activation of IL-1 beta, even if the ESC protein is not there and the inflammasome cannot be formed. However, I must admit that IL-1 beta was added manually to our experiment because although we confirmed its presence using Western blot analysis, well, we were not able to identify it using mass spectrometry analysis. Thus, it is important to find a new technique that will first enable us to find this IL-1 beta in our analysis and then understand how other proteins can activate it. So this is why we want to do proximity-dependent biotinylation in collaboration with the laboratory of Dr. Jean-Philippe Lambert, where we have here the immature form of IL-1 beta that will be coupled to a biotin ligase, an enzyme that uses biotin in the cell culture medium to capture proteins that comes close to the immature form of IL-1 beta, and it will bind them using strong interactions so that they cannot leave when they are close to IL-1 beta. And then we will purify this protein aggregate and identify its main component by mass spectrometry. Thus, this will enable us to identify, well, maybe known, but also new proteins that interact with the immature form of IL-1 beta and can activate it. Thus, we will try to inhibit these new proteins in an in vivo model of mice having the EAE disease, and hopefully this will lead to new therapeutic strategies against multiple sclerosis. So finally, I just want to thank my all laboratory teammates for their invaluable assistance through this big project. I also want to thank the following organisms: Multiple Sclerosis Society of Canada, CIHR, and NSERG to make this project possible. I want to thank the NeuroSymposium organizing team to make such an amazing event for us, and I want to thank you all for your attention.
So today I'll be covering the topic of our project, which is based on the development of a novel optogenetic-based model of alpha-synuclein aggregation to study Parkinson's disease. As you may know, Parkinson's disease is actually a neurodegenerative disorder characterized by the massive and progressive loss of dopaminergic neurons, as you can see here in the substantia nigra pars compacta, leading to a significant depletion of dopamine in the brain, which leads to the manifestation of motor symptoms including akinesia, muscle rigidity, tremor, and postural imbalance. So besides the neuronal loss that we see in PD, PD is also characterized by the presence of obviously in the remaining dopaminergic neurons, the presence of Lewy body structures, which are intracellular neurons called Lewy bodies, which are mostly comprised of alpha-synuclein. So more than a century since their first characterization by Dr. Frederick Lewy, little has been known about Lewy bodies and their role in PD, which is mainly caused by the lack of proper models to actually investigate alpha-synuclein involvement and Lewy body formation in Parkinson's disease. I present here a list of some of the models that have investigated the role of alpha-synuclein in Parkinson's disease. And as you can see here, the table has missing green boxes indicative of the lack of proper modeling. At first, with the identification of alpha-syn's involvement in Parkinson's disease, with genetic studies also confirming that the overexpression of wild-type alpha-syn or the mutated forms of alpha-syn can have a potential of recapitulating or representing some of alpha-syn aggregation propensity. However, none of the models were able to capture all of these features. As you can see here from this representation, when overexpressing alpha-syn, there's no inclusion formation. So there's no true recapitulation of aggregation formation in the cells. This is also evident in vivo as well. So this presents the lack of proper modeling and therefore lack of proper understanding of how alpha-syn is involved in the aggregation and PD pathology. So some of the features that are authentic to LBs include amyloid-like properties, forming beta sheet structures, the fact that they're detergent insoluble. They possess post-translational modifications, including phosphorylation and ubiquitination, presence of lipids and membranous organelles within the Lewy body structures, and presence of proteins implicated in cellular responses, such as HSP70. So faced with such limitations for the absence of cellular and animal models, it has become difficult to study the early cellular and molecular events related to protein aggregation, and it has also been difficult to monitor in real time the process of protein aggregation and inclusion formation. So this brought us to the idea of why not use optogenetics, which is now optobiology is being used very commonly for studying protein-protein interaction and protein aggregation. So we wanted to see if we can apply optogenetics to induce control alpha-syn aggregation in living cells. The advantages of application of optogenetics is that they're easy to obtain, they're non-toxic, and they provide with high spatiotemporal resolution for manipulation of in cells and in vivo. And additionally, they're highly tunable. So our project and our model is actually based on the application of Arabidopsis thaliana, which is a photoreceptor called cryptochrome 2 that allows for the proper clustering upon exposure to blue light. So we use this Cry2 system, which as you can see here forms robust aggregate formation upon light exposure. So what we wanted to do is make use of such application and fuse our alpha-syn to the light-inducible Cry2 system that allows it thereby promoting the formation of LB-like inclusions in living cells. So we engineered 3 constructs, including LIPA-empty, which is our control construct with the absence of alpha-syn, and our alpha-syn construct, which includes alpha-syn fused to Cry2 and M-cherry. And finally, another control we introduced is the use of the mutated form of alpha-syn, which is a non-aggregatable form of alpha-syn fused to the Cry2M cherry. As I've mentioned, upon exposure to blue light, this allows for the clustering of the Cry2 system bound to our alpha-syn. And notably, it's reversible. Upon inhibition of light, this system should be reversible. So here I would like to share with you some of the videos of live confocal imaging demonstrating the use and the real-time monitoring of protein aggregation using our LIPA system. So as you can see here, when exposed to light, we see a robust, fast formation of aggregates, which are characterized by inclusions formed in the cytoplasmic area. And here with the LIPA, we see formation of these inclusions, but I hope you have noticed the difference and the speed and volume of the aggregates observed in the LIPA-syn construct versus the LIPA-MT. And notably, in the LIPA-syn delta NAC, as you can see here, upon light exposure, we don't see any formation of these aggregates, suggestive that the LIPA construct is inhibiting the proper aggregation of the Cry2 system. So we next wanted to actually see if our inclusions can actually recapitulate many of the biochemical features of authentic Lewy bodies. So as you can see here, our LIPA-syn aggregates that are M-cherry positive, which are formed after light exposure, are positive for alpha-syn. They're also positive for the two main hallmarks of Lewy bodies, which is phosphocene and ubiquitin, which you see colocalization and presence of these markers. We looked also at thioflavin S, which is also another marker used to indicate the beta sheet structures, indicative of the formation of beta sheets in our M-cherry positive aggregates. Finally, HSP70 and P62 are heat shock proteins that are also been suggestive to be positive markers for Lewy body features. What was nice in our application is that what you see here is cells, hex cells exposed to LIPA-syn show authentic features, but we were able to recapitulate those features in a neuronal-based system, which are in this image. We capture all the authentic features in neurons derived from iPS cells. So this is a promising approach and showing the versatility of our application. We further wanted to look at the ultrastructural features that are characteristic of Lewy bodies. So we performed a nanoscale electron microscopy analysis, and what we saw from LIPA-syn aggregates from hex cells, we see the aggregates formed in the vicinity of the nucleus, but we also see a lot of proteins and vesicles surrounding the aggregates. And these were mainly composed of mitochondria and Golgi, but we were also able to see some forms of lysosomal and autophagosome-like structures. So what was interesting is that we looked at these and we wanted to further validate this using ICC examination from hex cells. And indeed, when we look at the cells, we see positive markers for the endosomal markers EAM1, the lysosome marker LAMP2, LAMP1, and the CD9 marker for all indicative of what has been recently suggested, that in Lewy pathology, in Parkinson's disease, the Lewy bodies are not only consistent of fibrils, but are also consistent of crowded organelles and lipid membranes. So our data also confirms the new emergence of the role of these and the presence of these organelles within Lewy bodies. So we next moved to in vivo to see if we can evaluate what we saw in vitro in vivo. And interestingly, when we perform AAV-LIPA introduction in our wild-type mice, concorded with the implantation of a wireless optogenetic light source to induce LIPA aggregation, what we see is that we were able to recapture all of the features using postmortem analysis, all confirmative of what we saw in vitro. Furthermore, we wanted to investigate the effect of LIPA-alpha-syn inclusions on dopaminergic neuronal loss. First, we confirmed that we have positive markers for phosphocene that are only present in the LIPA-syn and that are absent in the LIPA-MTU and the LIPA-delta-NAC, which are our two controls. So we confirmed the presence of true phosphocene LIPA inclusions. And when we examine the dopaminergic neuronal loss, we see that there is high and significant neuronal loss in the LIPA-syn injected mice that are exposed to the light, while when you look at the contralateral side, we don't see any loss. This is similar in accordance to what we see with the LIPA-MT and LIPA-delta-NAC exposed to light with no evident loss in dopaminergic neurons. So we further confirmed what we see here, which is quantified here in the TH neuronal loss in the Nestle test. The Nestle test further confirmed that it's actually indeed neuronal loss and not pertaining to dopaminergic loss per se. We then performed multiple behavioral analysis to confirm what we see in terms of dopaminergic neuronal loss. And we see that the alpha-syn aggregates actually induce Parkinsonian-like motor impairment, as evident with cylinder tests, which we see here. We see loss of coordination with the rotor test, and we see lack of grip strength as observed here, and also further confirmed with the gait test. So all these data are suggestive of the effect that the LIPA-syn have on a dopaminergic neuron loss, presented by the effect on behavioral compartments of the animals. So in conclusion, with building on the principle of optogenetics, we were able to develop a novel system that's capable of triggering the formation of authentic LBs, both in culture and in vivo. So the LIPA system has allowed us to induce alpha-syn aggregation in living cells with high spatiotemporal resolution, which is a very interesting feature available with using this system. The LIPA-induced alpha-syn inclusions actually recapitulate all the cardinal features of authentic Lewy bodies, both in culture and in vivo. And the LIPA-mediated alpha-syn aggregation actually precipitates the dopaminergic neuronal loss as evident in vivo. So I believe that our model can show you that we have been able to prepare to model a unique and reliable experimental model of alpha-syn aggregation that will allow us to study Parkinson's disease pathogenesis. So I'd like to acknowledge the work that I present here is actually a group effort from all members of our lab. And we also do acknowledge the involvement of all our collaborators from Université Laval, from McGill, and from Korea Advanced Institute of Science and Technology. And finally, I'd like to thank all the organizations funding and supporting our project. Thank you.